Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Rabbi shrah li sadri wa yassir li amri wa hlul 'uqdatam min lisani yafqahu qawli, amma ba'd. Guys, I hope semua orang sihat. Okay, masih lagi duduk di rumah mematuhi all rules for PKPPP, and let's continue our class. Okay, we're still in chapter 7, the last chapter, gene sequencing, and this is hopefully is the last class, the last ODL week, and maybe next week there will be no class at all, hopefully. Okay, so where we stop last week, we'll stop until automated. Yeah, this one. Right. So for last week class, I already explained to you about the basics sequencing methods, which is consists of two methods. The first one is maximum Gilbert sequencing. It's based on chemicals, and the second one is the Sanger method, with based on change termination sequencing. And nowadays, in lab, mostly in the lab, if you want to do a sequencing or you want to check your DNA sequencing of your recombinants, we mainly use modern automatic sequencing, where it's everything is already automated in one machines. So we just basically put your DNA sequence along with the ingredients, the NTP, so on and so forth, and you put it in the machine. Then the machine will read it automatically. So by the end of the day, you don't need to you don't need to do an agarose electrophoresis to check the patterns, because the machine is already read the sequence of your sample by color sequence. All right, so it used for type of fluorescent dyes to label each DDNTP. So in this modern automated sequence, we still use Sanger change termination methods, where DDNTP will stop in each reaction. So whenever DNA polymerase use DDNTP, the reaction will stop. And in this modern automated sequencing, to make it more easier, DDNTP is colored, means that labeled with a fluorescent label. So for example, A colored with pink, T colored with green, C colored with blue, and G is colored with yellow. So all four DDNTP can be added in one direction. The same goes to the Sanger method, basic Sanger method just now. Previously, cycle sequencing done just like in the tracer. Yeah, this one is method. Fragments separated in the capillary electrophoresis tube. The color label of each fragment is spread using a laser sequence generated using computer inference. So let's see. Okay, so basically this is what happened in the machine lah. Okay, so for example, this is your target sequence, unknown sequence. And when you add DNA polymerase, this is the DNTP plus limited amounts of fluorescency labels, DDATP, DDGTP, DDCTP, DDATP. So the DNA sequence stretch, new strand will grow like normal DNA lah. So whenever the polymerase use DDNTP, the elongation reaction will stop. For example, T here, so the DNA polymerase uses NTP, so it stopped. And the next one, the NTP will be used, and the next one again DD NTP will be used, and direction stops. So the elongation process is elongated like these ones, and each color patterns of the NTP is being detected by detection. So the fragment is read using a laser, where the laser will bombard the NTP, and the color will be read by a detector. So it's basically a color-based sequence lah. So instead of dia baca T T G T T P A C G G, so it gives you a color: yellow, green, blue, green, yellow, green, red, so on and so forth. So by this color, you can actually know the actual sequence of your DNA sample. Okay, so the color basically will be sequenced from 3' to 5'. So we have to convert it, okay, to make 5' to 3', which is the leading strand lah. Okay, so basically the DNA sequencing will give you a graph result like this. Okay, a colored graph result like this. So for example, like this. So this is the DNA sequencing results, actual results. So the sequencer will give you two types of results. The first one is graph like this, right? And the second one is in a full sequence stretch, so without a graph, so only a sequence. And another one is the graph like this one. So why it have two sequence? The first one, it's the important one, is this graph lah, because from this graph you can know is there any problem in sequencing. Okay, so is there the nucleotides, is the position of the nucleotide is correct or not, based on this graph itself. So for example, okay, kalau you tengok the graph is spiking like this, right? Spiking, means that if the graph spike is higher, so tinggi, for example, yang ni contoh yang ni lah, right? So tinggi, so and sharp, right? It have to be sharp. Sharp, it means that in this location confirm adalah T. Okay, so ini pun also adalah G, right? So this is kita cerita pasal yang tinggi punya lah. So it confirm like maybe like 80% or 90% is the exact nucleotide position here. Okay, kalau yang rendah-rendah ni, spike yang rendah-rendah ni, okay, you can boleh assure juga yang the positions of nucleotide is correct. For example, this one is C, ini adalah A, ini adalah G. So based on the graph, walaupun dia pendek, tapi graph the spike dia tajam ke atas tu, means that yeah, surely itu adalah position yang betul. But then kalau you tengok yang belah-belah bawah ni kan, so macam it's like a noisy signals, right? So yang rendah bawah ni, for example, I ambil yang bawah ni, right? Okay, yang bawah ni, the top of sini ada two peaks, alright, two blue peaks, which is blue apa? Blue C. Okay, C. So yeah, C. So blue is C, give like two peaks, alright. So peak ni dia tak separated and tak spiking like the previous one. So ada dua peak. So even though it give a correct sequence here, C here, but then you can't confirm it like 100% confirm that this position is belong to C. Okay, so you have to double check it with a sequence alignment lah. So kalau you ada sequence recombinant U, gene of interest U, and you buat recombinant, then you nak check date lepas recombinant. Sometime after you make a clone DNA recombinant, the base is mutated. So we want to avoid those mutations to be appear in our DNA recombinant. So that's why we do DNA sequencing after cloning just to ensure that gene of interest kita dengan after recombinant is aligned, multiple alignment. Kalau yang rendah-rendah macam ni, you have to check back lah. Okay, usually yang rendah, yang graph yang rendah ni appear during the earlier stage of elongation. So primer, for reverse primer, forward primer. So at this region, usually graph tu tak cantik lah sebab dia baru nak start. And also bila nak habis. So bila nak habis pun graph tu memang takkan cantik. Okay. So in the middle, the graph should be look like this. So kena cantik lah. Right, to ensure that correct position of your nucleotide sequence. Right. So an example tadi. Okay. So the Sanger method generates sequence of about 900 base pair, of which 500 base pair are good to reach. Okay, so dalam Sanger method usually the maximum stretch yang DNA polymerase can go forward and reverse it's only about 900 base pair lah. So reverse 900, forward 900. So in between tu dia align, dia akan cross over lah. Okay, but then dalam 900 tu hanya 500 saja yang are good to reach. Good to reach means that the spike is like this. Right? Cantik tajam. But then 200 in front and 200 starting sequencing and 200 yang ujung tu memang tak cantik. Alright? Dia akan jadi macam ni. Okay? Even though sequence tu betul, tapi kita tak boleh nak 100% percaya. Right? So that's why kita kena buat primer tu. Kalau DNA sample you pendek, like 500 base pair ke atau 600, 700 base pair ke, tak jadi masalah. You nak buat sequencing. Okay? But then the human genome is actually 3 GB, terlampau panjang, billions of genes, human genome. Even a small bacteria, we have some genome of several MB. Okay? So how to sequence such a long piece of DNA? So this is the problem lah. So since in Sanger method, dia hanya dapat buat 900 base pair and only 500 base pair are good to read. But then imagine if human genome or bacterial genome yang ada besar, right? The punya size of the DNA is too big. So how do we read this one? Okay, so there is a 2 methods to read the big genome, the big size genomes. The first one is a back-to-back assembly, and the second one is whole genome shotgun sequence, WGS. Okay, so okay. So ada 2 method, back-to-back assembly and WGS. The first method is back-to-back. Back is actually means bacteria artificial chromosome vector. Okay, so when you heard about back, it will relate to back library. Okay, main Z dia macam ni. So for example, chromosome tu panjang, alright, 3 GB. So we will use a specific enzyme to cut the chromosome sequence, right, into a pieces. So bila kita guna enzyme tu tadi, it will cut automatically of the long DNA sequence, make it shorter. Okay, so back ni is a vector that can hold a large sequence, for example, a sequence of DNA, for example ni daripada 150,000 base pair. So kromosom yang panjang ni bila cut, so sequence yang banyak ni the maximum it can go is 1,500, 1,050, 1,050 base pair. 1,150 base pair. Okay, so the short sequence ni tadi, not like short sequence, still long, but then it can be clone into a back vector. So bila dia clone dalam back vector ni tadi, okay, ni segment 1, segment 2, segment 3, segment 4. So actually we create a back library. So this back vector, after we clone, kita akan bubuh dalam E. coli lah, mostly dalam E. coli, to amplify it, untuk simpan as a stock. So that's what we call a back library. So senang kita nak refer balik. Okay. So kita tahu, okay, segment 1 ada dalam bakteria ni, segment 2 ada dalam ni, dan ada dalam ni. So everything are in order, ada data kita. Macam library lah. You pergi library kan, nak pinjam buku, you tanya kat librarian tu, okay, nak pinjam buku ni, nak scan, after by scan. So dia check dekat dalam database dia, okay, dekat rak nombor 12. So you just go at rak number 12 and you can find the book. Sama juga macam back library ni. Okay. So you tahu, okay, fragment number 1 dekat dalam bakteria mana? Okay, bakteria ni simpan kat mana? Kat sini, kat sini. So you tahu Okay. So from this back library, okay, this is the more details one lah. So chromosome, for example, chromosome number 3. So potong. So for example, dia ada 500 fragments. So 500 fragment ni tadi dia akan masukkan ke dalam back vector lah. So each fragments we call it counting maps, right? Counting maps. This is one counting fragment, counting, right? So on and so forth. Okay, so ni sama juga. Kalau you nak buat guna M13 vector yang besar tadi, boleh potong cut out lagi to make it shorter. You boleh masuk dalam M13 vector. Okay, so this is M13 vector. Ini nak pakai ni lah, senang sikit. Alright, okay. So this is the summary lah of the back-to-back punya method. So this is the whole genome of DNA, and you cut with a specific enzyme, cut until you have given short fragments, and you make a back library. You make a back library means that you clone masuk dalam back vector and you transform masuk dalam E. coli, amplified, then you simpan stock. Which fragment is which, you tahu. Okay, that's one is back library. So after back library, you create a counting maps. So each fragment tu tadi, you hantar sequencing. Alright, sequencing by Sanger method, automated Sanger method. So yang panjang tadi kan, so for example 150,000 tu tadi, terlampau panjang, so it cut more shorter masuk dalam M13. Right, still considered as a back library lah. So after that, yang short fragment tu tadi, you will create a counting map. So counting map ni of the sequence lah. So sequencing this fragment, this fragment. So you do a sequencing by a fragment. Actually dalam back to back and WGS ni, there is no how to say a new method actually. It still use automated Sanger sequencing, but then why we call shotgun, because at this part we do a sequencing by fragments. Okay, so after a complete fragments, kita try to align. Okay, fragment ni ni sequence dia, fragment ni sequence, then we put this results in one database, one computer, and the computer will run a software that can detect which part is aligned together. So for example, fragment number 1, this is fragment number 2. So bila dah buat sequencing, automatic sequencing, kita dah ada sequence DNA dia. Then when we align, actually, for example, hujung fragment A ni, hujung fragment nombor 1 ni actually align dengan hujung start of fragment number 2. So align means that dia punya sequence sama. Right? So kita tahu, okay, ni sequence yang betul, align. Then after that, fragment number 2 align pula dengan fragment number 3, so on and so forth. So that's approach we call a shotgun approach. Sequence is counting by a shotgun approach. So this is what we call a shotgun approach. Okay. So bila kita dah ada banyak, okay, for example ni, fragment number 1, this fragment number 2, fragment number 3, they align. Okay. And fragment number 3 juga yang kat tengah ni pula, sometimes ada pula align with another fragment, short fragment lah. For example, this one, okay, adalah dekat here, the position is here. Then we can generate the full sequence of the DNA lah. So tak kisahlah DNA itu panjang mana pun, kita still boleh buat sebab apa, kita potong using a enzyme. Okay, this is back-to-back method. And the next method is the whole genome shotgun sequence method, WGS. Okay. Same approach. They use a shotgun where it relies on the alignments of the DNA. But then in this time, dia tak buat back library. Okay, lagi cepat lah, means that lagi cepat, tapi you tak ada stock. So for example, you nak check lagi fragment number 1 tu macam ni, kalau kat dalam back library you have the stock lah, right? Mana kat dalam whole genome ni you tak ada stock. You just cut using mechanical shredded DNA, tak pakai enzyme, you potong je. Potong. So jadi small fragment ni tadi, directly you buat sequencing. Okay. So from that sequencing, kita pakai shotgun approach sama juga. Okay, fragment number 1, fragment number 2, fragment number 3, they align together. And daripada alignment tadi kita tahulah dia punya susunan sequence dia. Okay. So generate a finished sequence. So this is an example of WGS sequencing method. So both methods are basically used in the Human Genome Project. But to Human Genome Project tu masa yang sangat panjang. Sebab apa? Sebab dia pakai back-to-back library method. Okay, so Human Genome Project dia kena cut, and then each fragment dia kena clone masuk dalam back vector, simpan, and read one by one to get the whole sequence. Okay, so back sequence is slower and more expensive, of course lah. Okay, kalau you tengok Human Genome Project, dia yang kawan you do a video on that. It took like berapa puluh tahun untuk complete the whole Human Genome Project. So very slow, but it will create less error and easier to assemble. Sebab apa? Sebab you dah ada stock, you make stock. Okay, kalau ada problem, you can always go again and refer to that particular fragment yang dah clone tu tadi. Okay? And shotgun sequencing is much faster. Alright, WGS is much faster. No need to make a counting map. So tak payah nak buat counting map, kita punya WGS lah. But then the counting map yang kita clone masuk dalam bag tu is a slower step. Actually, yeah, it's very slow because you have to clone one by one, and you have to make a stock one by one. So banyak, just imagine how many sample you have. But cannot be used to sequence genome with a high amounts of repetitive sequencing. Yes. Okay, kalau you pakai WGS, kalau ada C, tandem repeat, you ada G, ada so on so forth, so memang susah. Sebab habis tak tahu mana satu yang hujung, mana satu yang depan. Okay, sebab semua sama, line together je, semua sama, saya tak tahu. Kalau for example ada G, ada C semua, so dia akan align, stacking. So you tak tahu which fragment is where. And so susah lah if the sequence is in repetitive sequence. And more prone to errors in many sequence have to be matched and assembly blindly, of course. Apa you tak because it cuts by shred DNA mechanically, you tak pakai enzyme, just cut blindly lah. So it will increase the error of the sequencing later on. Okay, it will affect the results. Right, then next is next generation sequencing. They need to rapidly sequence a lot of DNA, new technique in sequencing. So kita ada traditional method, kita ada advanced methods of DNA sequencing. Kalau traditional tu Maxim Gilbert dengan Sanger method, and advanced is WGS and back to back. And we have another one, the third one is next generation sequencing. It's more powerful techniques. Very high throughput, many thousand of DNA strands sequenced together at the same time can sequence millions of bases per day. So this is example of the NGS sequencing lah. Ada linear sequencing, ada Pyro Sequencing, ada IO torrent next sequencing, ada bisulfite sequencing. So this one I think ada 4 slide je, so you can read on your own. I can explain briefly only. So the first one is Pyro Sequencing. So Pyro Sequencing is actually the principles of Pyro Sequencing is based on lights. This light is actually generated from ATP and luciferase. So kat sini kan, kalau you tengok dekat ni, DNA, N ni lah bilangan nucleotide tu tadi. So bila DNA ni is elongated and we use DNTP, so for DNA N plus 1, so 1 base dah masuk. So each base that added into the sequence, it will release a PPI. PPI is pyrophosphatase, right? So pyrophosphatase ni tadi akan use to generate ATP, right, from APS. Okay, so kalau you tengok nanti kat sini, kat dalam slide you tak ada dia punya reaction mechanism, you boleh cari kat Google or YouTube. So APS yang tadi, okay, akan dipakai oleh PPI. APS along with PPI akan generate ATP. So ATP ni tadi lah akan change the luciferase into a oxyluciferin that will emit a light. So the light ni tadi akan read by a photodetector. Okay, so dia akan A akan keluarkan light sebagian, T another light, so it akan read the sequence light. Okay. And then the next one is bisulfite sequencing. So bisulfite sequencing is actually the principle behind it is read the methylated nucleotide. So methylation, CH3 lah. So each methylated nucleotides can be read by the detector. So methylation yang CH3 tu lah. Okay. So there is a 2 step, which is the first one is bisulfite conversions and another one is bisulfite sequencing. So what happened in bisulfite conversions? So kat dalam nucleotide sequence tu only cytosine is methylated. Okay, so bila sequence cytosine tu methylated, it will carry how to say apa term tu, okay, macam ni I explain to you the methylation. So you have 2 mouse, okay, mouse 1 and mouse 2. So mouse 1 color is white and another color is grey. Okay, but then the DNA sequence of both mouse is 100% identical. Okay, but then you tahu yang gene tu tadi akan affect the phenotypes, betul? So putih adalah phenotype color putih, green adalah phenotype untuk mouse number 2. Even though the DNA is 100% identical, supposedly color should be same. So then, but then it is possible the phenotype of two identical DNA sequence can be crossover, boleh bertukar. By why? By methylations. Okay. So kalau cytosines in DNA yang white ni tadi tak methylation, it give you white lah. Tapi kalau cytosine yang dalam grey mouse ni is methylated, tambah CH3, so it give you a grey color. So it can be changed lah, even though the two identical DNA. Okay, so dalam the first step with bisulfite conversion, cytosines yang tak methylated will be changed into uracils. Whereas the methylated cytosine is not changed by bisulfite treatment, so bisulfite conversion lah. So kalau C tu tadi, cytosine tadi ada dah siap-siap methylated, dia takkan ubah. It's still methylated cytosine. But then ada yang C tak methylated, it will change to uracil U. Okay. Then bila dia nak sequence, okay, for example, C is methylated. But then C ni tak methylated. So bila kita buat treatment of sequencing, bisulfite conversions or bisulfite treatment just now, yang C, the unmethylated C tadi will be changed into U. Right? So we know later on yang position C ni tadi is not methylated. Kan? So bila kita when it change to U, this one change to U, then the untreated reference is like GTC, this is the methylated one, GG methylated one, C methylated one, GATC gomet yang ni untreated. Okay, the treated one, GTC. Okay, we know it remain the C, means that the C is methylated. GGC methylated. GATU. So the C change to U means that the positions of C here is not methylated. So that's why ditukar kepada U. C, this is methylated and this one is not methylated. This sequence indicate that the C's are methylated. Yang ni methylated lah. This one is not methylated. Right. So this is basically the summary of this chapter. If you have ada doubts, tak faham tentang bisulfite sequencing or Pyro Sequencing or automated sequencing, you can refer me, you can text me anytime. Or you can go through YouTube lah. There is a lot of animations to explain how the principles behind each sequencing methods. Okay, so I think we stop until here. Alright, this is the last chapter, this is the last class, but I want to remind you that this week's on 26 kita ada test number 2. Alright, and on 29 you have to submit your group assignments. Alright, in group assignments, you just upload the video on the IG that are already been created. Macam last time lah, you post in individual assignment. So I think there is, kalau everyone want to send me directly, okay, sometimes ada video yang dia punya size terlampau panjang kan, terlampau besar, so susah nak transfer, susah nak send directly. So you just upload it on the Instagram, then I will check through Instagram lah. The length 5 to 8 minutes, I think 8 minutes terlalu panjang lah. 5 to 7 minutes ke 5-6 minutes ke is enough lah for one video. Okay, so on 29 don't forget to upload it on Instagram, then I will check it on your video later, then I will evaluate your each video. Okay, so I think that's it. Okay, kalau any question, you can directly text me and ask me apa yang you tak faham lah. Okay, okay guys. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.